Let's now move on to the final type of carrier that we're going to talk about in this lecture, and that's solute carriers. The prototype that we're going to use is an SGLT1. These particular transporters are very nice, and in this particular example, we're going to have a molecule that will enter into this particular cell, into this transporter, and then have it ratchet to exit. So first the solute binds within the carrier itself in a binding site, and then finally it changes, or there's a conformational change that happens that then allows it to enter into the cell. Now, how the solute is released is also important. So it not only needs to bind, but the carrier needs to have that change occur, and then it also needs to open up on the aft side to allow it to move through the membrane. What do you need for a solute carrier to work well? The big thing is you need a concentration gradient. So similar to a pore and similar to an ion channel, you need to have the concentration gradient developed already. The other thing that you need is to make sure that the transport substance, does it transport by itself or does it need a co-transporter molecule? For our particular prototype, we are going to use one that requires a co-transporter or another transporter that's being brought along. If the solute is brought along in the same direction, it is called a symporter or co-transporter. If it involves a substance in which it will go the opposite direction, it's called an antiporter or countertransporter. Or finally, we have things that just move through by themselves, or uniporters. So why have a solute carrier in the first place? Why not use simple diffusion to move a molecule through the cell membrane? Well, first is it might not be able to make it through the cell membrane if it doesn't have enough of a nonpolar or lipophilic nature. So that's one reason. The second is you can move things through faster with a solute carrier than you can with simple diffusion. So if you look at this particular graph, you have concentration of the substance along the x-axis, and along the y-axis you have the rate of diffusion. Focus in here on the red line, the facilitated diffusion line. You can see how it occurs at a lower concentration. You get more rate of diffusion with facilitated diffusion through a solute carrier than you do through simple diffusion. So it's faster. The one problem that can occur is that sometimes, besides it being faster, you can do it at a lower concentration, meaning that you need less of the substance available for that fast transport. This is very important in terms of getting things through as rapidly as possible. The one problem with a solute carrier is that you only have a certain number of solute carriers, and eventually you can eclipse the number of carriers that you have available. And if you do that, you can then have the substance spill over. And this sometimes occurs in places like the kidney, which we'll get to in a few slides. Now, the prototype that we want to focus on today is the SGLT1, which is the sodium glucose cotransporter 1. And this is one of the primary ways in which you're going to bring glucose into the body in the GI system. So this is usually how it occurs if you take in, let's say, milk or some sort of a dairy product. You have lactose, it gets broken down by an enzyme called lactase into glucose and galactose. Now both of these particular carbohydrates, simple carbohydrates, are going to need a transporter to get into the body. SGLT1 provides that transporter. In this case, we're going to be moving in one glucose molecule and two sodiums. What do you need to have this process work? The first is you need to have a concentration gradient. So sodium needs to have a concentration gradient to move in glucose into the body. How does that concentration gradient get developed? It's through another transporter. The sodium-potassium ATPase, which is located on the other side of the enterocyte, on the basolateral membrane, is creating the gradient, so you need to pull sodium through the cell. And then the sodium is providing the driving force to move that particular glucose or galactose molecule through the transporter. It's interesting, for SGLT1s, you actually need 2 sodiums. For SGL2s, which is another transporter in this particular family, you only need 1. So it depends on the transporter which molecules are going to be brought through it. But both of them are called symporters or co-transporters. Now this allows for glucose absorption, and the example that we're using with this particular SGLT1, it's through the small intestine to allow us to reabsorb glucose into the body, and that allows us to have enough energy for ATP production. There are other SGLT1s in the body besides those that are located in the GI tract. One of them is located in the renal tubule, and this allows us to reabsorb glucose so we don't lose it out of the body. And so in this case, we're moving glucose from the tubule lumen space in through the renal cell into the blood. And in this case, we really want to make sure we don't lose glucose out of the body. So it's a very important transporter. However, if a person is diabetic and has a high amount of blood glucose, you could eclipse the Vmax of the SGLT1 within the renal tubule cell, and therefore glucose will be spilt over into the urine and you will urinate it out. In this case, you'll be losing glucose in a diabetic hyperglycemic condition.